Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas with your tutor Dr Shehla Aman. In today's class of Histology of Nervous System. So in today's class we are going to discuss the different types of the supporting cells of the nervous system and discuss the histological features of all types of the supporting cells of nervous system. Supporting cells of nervous system are found in both the central nervous system and in the peripheral nervous system. They perform a supporting function for the neurons. In central nervous system, they are called the neuroglial cells or simply the glial cells, and they are the oligodendrocytes, the astrocytes, the ependymal cells, and microglia. And in peripheral nervous system, the supporting cells of the peripheral nervous system are the Schwann cells and the satellite cells. So first we will discuss the Schwann cells. The Schwann cells are the types of the cells which myelinate. They wrap around the axon in the peripheral nervous system during the fetal development. Schwann cell cytoplasm and nucleus form outermost layers of the neurolemma, with the inner portion being the myelin sheath, and tube guides growing axons that are repairing themselves. So in this diagram you can see this is the axon and this is the Schwann cell. It's a cytoplasm and its nucleus. So initially this axon is present in a groove formed by the Schwann cells, and when the edges of the groove meet and they fuses in the midline, and then they starts to wrap around the axon, so they form the myelin sheath. So oligodendrocytes myelinate the axons in the central nervous system, and broadly the flat processes wrap around the central nervous system axon, but the cell bodies do not surround the axon. So the difference is that there is no neurolemma is formed, and little regrowth after injury is possible due to lack of the distinct tube or the neurolemma. So what is the process of the myelination? So first you can see, look at this diagram. This is the axon and this is the Schwann cell which is forming the myelin sheath around the axon. So they form myelin sheath around the peripheral axons, look like jelly roll with numerous neurolemma covers, and node of the Ranvier separates each Schwann cell. So in this diagram you can see first it is present in the edges in a groove formed by the Schwann cell, and when the edges of these neurolemma, the Schwann cells cell membrane meet, they form the inner mesoaxon, and then they starts to myelinate. So during this process of myelination, these Schwann cells wrap itself around the axon several times, and its cytoplasm is extruded out of this myelination process, but at certain places you can see this cytoplasm as well. So during process of this myelination you can see the nucleus is present at the outer collar, and these all are the myelin sheath, which are the lipid rich material whose purpose is to myelinate and to insulate this axon. So this is the process of the myelination of the peripheral nerve, and in this you can see this is axon and this is the inner mesoaxon which is forming. When these inner two leaflets fuses, they form the inner mesoaxon. Now the process of wrapping continues. You can see in this diagram, and cytoplasm is extruded out. So cytoplasm which is present initially was inner collar of the cytoplasm, and cytoplasm which remains outside will form the outer collar of the cytoplasm. So in this diagram you can see this is the axon which is present, and inner leaflet meet with each other and they form the inner collar of the Schwann cell cytoplasm. Here you can see this is the outer collar of the Schwann cell cytoplasm which is present, and this is the nucleus of the Schwann cell. So this is the photomicrograph of osmium fixed talvidine blue stained section of the peripheral nerve. This is cross section and this is the longitudinal section. So in this cross section you can see this clear area is axon, and this dark blue area is the myelin sheath which is formed around this axon. In certain section you can see the two rings are present. Here you can also appreciate the two rings. So these are the section which are taken from the area where Schmidt-Lantermann cleft is present. This is the longitudinal section, and here you can see the node of the Ranvier is present. Also you can see the perinodal cytoplasm is present. This is the Schmidt-Lantermann cleft, and these are the myelin which are formed by the Schwann cells. During the process of the myelination, there are certain terms which I am repeatedly using. So what are these terms? Now let's explain these terms. First is the major dense line. So you should remember that when this axon is present in a groove of the Schwann cell, and when the Schwann cell's membrane meet and fuses, they form the inner mesoaxon. So I can explain this also in this diagram. These are the inner cell membrane of the Schwann cell, and this is the outer membrane. So when these two inner membrane fuses with each other during the process of the wrapping, you can see in this diagram, also you can see in this diagram. So when these two inner leaflet meets, they create a darker area line which is more dense, electron dense, and this is called the major dense line. Also you can see when these outer leaflet meet with each other and they fuses, they then create a light color lines. In between these darker lines you can also see the lighter lines which are present, and these lighter lines are called intraperiod lines. So please remember, the major dense lines are formed when outer leaflet fuses with each other and they are more electron dense. That's why they appear darker, they appear opaque, and these are the major dense lines. And when these two outer leaflets of the plasma membrane fuses with each other during the process of the wrapping, they produces less dense lines called the intraperiod line. Next, different Schwann cells wrap around the part of the axon to form its myelin sheath, and they spiral around a short segment that is 0.08 to 0.1 mm, and at the junction where the two Schwann cells meet, there is the node of the Ranvier is present. This is the area where the myelin sheath is not present, and this is important for saltatory mode of the conduction of the nerve impulses. So now I further explain the other terms. So look at this diagram. This is the axon, and these are the different leaflets of the Schwann cells which are forming the myelin sheath. The darker one are the major dense lines, and the lighter ones are the intraperiod line. So this area is the node of the Ranvier where two Schwann cells meet. Now remember, when the process of the myelination take place, the cytoplasm of the Schwann cell is extruded out during this process of the wrapping. Only a small part is present close to the axon, and this is called the inner collar of the Schwann cell cytoplasm. So that is the inner collar of the Schwann cell cytoplasm. Also at certain places in between there is some collection of the Schwann cell cytoplasm, and these are called the Schmidt-Lantermann clefts. Then the cytoplasm most of the cytoplasm is extruded out, and it is present outer to the myelin sheath, and it is forming the outer collar of the Schwann cell cytoplasm. So these are the outer collar of the Schwann cell cytoplasm where you can see the nucleus of Schwann cell as well. This is the inner collar of the Schwann cell cytoplasm, and these are the Schmidt-Lantermann clefts. Also at the node of the Ranvier there is a small amount of cytoplasm present at where this is called the perinodal cytoplasm of the Schwann cells. So also remember, during process of fusion and wrapping, when inner leaflet fuses, they form the inner mesaxon. Also during when the process of myelination is completed and to close the groove and two outer leaflet fuse it with each other, they will form the outer mesaxon. So these these are the different terms which are used, which I am repeatedly telling you about during the process of the myelination. So this is again inner collar of the Schwann cell cytoplasm. These are Schmidt-Lantermann cleft. This is outer collar of Schwann cell cytoplasm. This area is the perinodal, and this is the node of the Ranvier where axon is present. So if we hypothetically uncoil a Schwann cell, hypothetically if this is the axon and this is the cytoplasm of the Schwann cell which wrap around around the axon. So if we hypothetically uncoil the Schwann cell, we can understand all of the terms which are using. So this area is the inner collar of the cytoplasm which is present close to the axon. These areas of the cytoplasm which are wrapped, which are present in between the wrapping and the myelin sheath, these are called the Schmidt-Lantermann cleft, and this is the outer collar of the Schwann cell cytoplasm where you can see nucleus as well as subcellular organelles of the Schwann cell. So this is the process. If you hypothetically uncoil Schwann cell, you can understand all of it. Now this is a beautiful diagram, electron micrograph of an axon. So this is the axon in which you can see different microtubules as well as mitochondria which are present, and this is the inner mesoaxon, and outer to this inner mesoaxon small amount of cytoplasm which is present. This is called inner collar of the Schwann cell cytoplasm. Then these all are the myelin sheaths which are formed by the Schwann cell, and this is the Schmidt-Lantermann cleft, and then this area is outer collar of the Schwann cell cytoplasm which is present. Also here you can see outer collar of the Schwann cell cytoplasm. This insert show enlarged area from this area. This is the enlarged photomicrograph, and in which you can see the darker lines, the major dense lines and the interperiod lines. Also you can see the Schmidt-Lantermann cleft which is present. This is another beautiful diagram from the unmyelinated axon in the peripheral nervous system. So this is the Schwann cell. Unmyelinated in peripheral nervous system doesn't mean it is truly unmyelinated. It is present sometimes in grooves of the Schwann cells, but the edges of the grooves are not closed and they are exposed. That's why it is called the unmyelinated. So this is the diagrammatic presentation, and this all is the electron micrographic picture in which you can see different axons which are present in the grooves of the Schwann cell, and this is the nucleus of the Schwann cell. So again you can say this is the neuron, its a cell body, it's a nucleus, its subcellular organelles, the axon hillock, the initial segment, then the axon, its main branch, its collateral branch. This is the node of the Ranvier. This is the myelin sheath which is formed by the Schwann cell. Also you can notice the Schwann cell nucleus which is present over here. This all is the axon, and this is the peripheral process which is forming the axon terminal and synaptic end bulb. This is H&E stain section taken through the cell body of the neuron, in which you can see prominent nucleus and nucleolus giving you the owl's eye appearance. Also the nasal substance is present, and these are different dendrites and the axon which is present. Myelinated and unmyelinated, you can compare both in this diagram. So this is electron micrographic picture again, and in which you can see this is the axon and this is the myelin sheath which is present around the axon, and this is the Schwann cell cytoplasm and its nucleus, and this is the basal lamina of the Schwann cell. In this you can see this is the unmyelinated axon which is present in a groove of the Schwann cell, and this is the neurilemma, and this groove and this process of wrapping is incomplete. So myelination in peripheral nervous system is that all axons surrounded by lipid and protein covering myelin sheath produces by the Schwann cells. Neurilemma in the cytoplasm and the nucleus of the Schwann cell, and gaps called node of the Ranvier. So they may be unmyelinated or they may be myelinated as well. Now we move on to next type of the cells which are present in the peripheral nervous system, and these are the satellite cells. Satellite cells, like Schwann cells, they are derived from the neural crest cells. So please remember, this is again your MCQ, that both the satellite cell and Schwann cells are derived from the neural crest cell. They surround the neuronal cell bodies present in the ganglia. So this is the neuronal cell body which is present in the ganglia, and these all are the satellite cells which are present around these neuronal cell bodies. They provide nutrient, they remove metabolites. They are the small cuboidal cell surrounding the neuronal cell bodies in the peripheral ganglia. They support the neuron in peripheral nervous system. They also originate from neural crest cells, and they establish and maintain the controlled microenvironment. So these are the satellite cells. They are present in ganglia. They surround the neuronal cell body. They are derived from neural crest cells. They are simple cuboidal in nature. Again, look at this diagram. This is the neuronal cell body, and surrounding is the satellite cell which is present, and this is the axon, and this axon is myelinated by the Schwann cells. So this is the H&E section of the ganglia, in which you can see this is the neuronal cell body, and these small cuboidal cells which are surrounding it are the satellite cell. In this diagram also you can see these are the small cuboidal cells. These all small cuboidal cells are the satellite cells, and these are the axonal processes which are coming out of these neurons. This is again another diagram at different magnification, in which you can see these are the ganglia. This is the diagram, is H&E stain section of the ganglia, and this is higher magnification, in which you can see the neuronal cell body along with the nucleus and the prominent nucleolus, and they are surrounded by the simple cuboidal cells which are the satellite cells. So this is the section of the neuron, and these are the different neurons which are present in the ganglia. These areas, please look at this area. So these are the axons which are present, which are coming out of these neuronal cell bodies. Again, another H&E stain section, in which you can see the ganglia, the neuronal cell body, which are surrounded by the satellite cell, and this is the same slide at higher magnification. So now we move on to the cells in the peripheral. We complete it. This is the satellite cell. This is the posterior root ganglia, in which a neuronal cell body is present, which is surrounded by the satellite cell, and this is the axonal process which is covered by the myelin sheath of the Schwann cell. So this is another diagram in which you can see different types of the cells which are present in the central nervous system, and these are cells which are present in the central nervous system. These are the ependymal cells, the astrocytes, the microglia, and the oligodendrocytes which are present. So we discuss now these cells one by one in detail. First, neuroglial cells. Half of the volume of the CNS is formed by the neuroglia, are simply the glial cells. They are smaller than the neuron. They are 50 times more numerous than neuron. These cells can divide. There is a rapid mitosis in tumor formations called the gliomas. 4 types of cells in central nervous system: the astrocyte, the oligodendrocytes, the microglia, and the ependymal cells. So in this diagram you can see these are first the ependymal cells which are lining the fluid filled spaces in the central nervous system. This is the oligodendrocyte which is forming the myelin sheath in the central nervous system. These smaller cells are the microglia, and this is the neuron, and this cell is the astrocyte which is present either in close relation to blood vessel forming the blood brain barrier, are in close relation to the neurons, maintaining its metabolic environment. So different types of cells: the ependymal cells lining the fluid filled spaces, the microglial cells, the astrocytes, the neuron, and this is the capillary, and this is the oligodendrocyte. So now we first discuss the oligodendrocyte. Oligodendrocytes form myelin sheath in the central nervous system, fewer branches than the astrocytes. So you can see this is the oligodendrocyte and it's a nucleus, and it is wrapping around multiple axon at the same time. So the process is different from that of the Schwann cells, because in case of Schwann cell in peripheral nervous system, Schwann cells wrap itself around a single axon, while in case of oligodendrocyte is present at its place. It sends tongue-like processes, and these tongue-like processes wrap around the parts of the axon. So multiple axons are myelinated at the same time by single oligodendrocyte. So this is the difference. These oligodendrocytes are most common glial cells which are present. Each form myelin sheath around more than one axon in the central nervous system. So this is the oligodendrocytes, and you can see this is forming myelin sheath around multiple axons, and it is analogous to the Schwann cell in the peripheral nervous system. So what is myelin sheath? This is the insulating layer around the nerve fiber formed by oligodendrocyte in CNS and Schwann cells in PNS, composed of lipoprotein with phospholipids, glycolipids, and cholesterol. Myelination is the process of formation of the myelin formation. Myelin allows nerve conduction to be 150 times faster than non-myelinated fiber. This occurs by saltatory conduction, and impulse jumps from one node of Ranvier to another node of Ranvier. So oligodendrocytes in this diagram, you can see this is beautiful diagram, the three-dimensional view of an oligodendrocyte as it relates to several axons. So this is the cell body of the oligodendrocyte. It is sending multiple tongue-like processes. You can see all of these processes, and these processes are wrapping around axons. So this single oligodendrocyte, its cell body is present at its own place, but its tongue-like process at same time they are forming myelin sheath around the axon in the central nervous system. Also remember all of the structure, the relationship of cytoplasm and myelin is essentially same as that of the Schwann cells, meaning that it has inner collar of the cytoplasm as in peripheral nervous system. It has Schmidt-Lenterman cleft. It has outer collar of the cytoplasm and also the perinodal cytoplasm. Also you can see during process of myelination it is forming major dense lines and intraperiod lines as well. So next is the microglia or microglia, and these microglia are the smallest of the cells in the glial cells. They have a phagocytic role. They are one of the cells which are derived from monocyte macrophage system. This is again your MCQ as well, and also as they are derived from monocyte macrophage system, so they reveals numerous lysosomes, inclusions and vesicles. They have little rough endoplasmic reticulum, few microtubules. Usually their amount is very less in central nervous system, but they increases in cases of certain inflammation of the brain tissue. So these have thorny bushes in appearance, and the smallest of the glia, you can see this is the microglia cell, phagocytic and originate from the monocytes. Next are the astrocytes. Astrocytes are largest of neuroglial cells. So you can see this is the astrocyte. They are star-shaped cells. They form the blood-brain barrier by covering the blood capillaries. So I have told you as previously that either these astrocytes are present in close relation to blood vessels or to the axonal process of the neurons. So where they are present in close relation to blood capillaries, they are maintaining the blood, they are forming the blood-brain barrier. They metabolize the neurotransmitters, they maintain the microenvironment of the neuronal cell bodies. They have prominent bundles of glial fibrillary acidic protein, which is most numerous in fibrillary type of the astrocytes, and they provide the structural support to the neurons. So astrocytes, again a beautiful diagram. This is the fibrous astrocyte. It has many processes either in relationship to the blood vessel where it is forming the blood-brain barrier, or in relationship to the axon where it is maintaining the microenvironment and giving support to the axon. So astrocytes are star-shaped, most numerous, and forming the blood-brain barrier. They are mainly two types, the protoplasmic and the fibrillar. So protoplasmic are having granular cytoplasm, have many branches and short processes found mainly in the gray matter. And fibrous are present, they have long slender processes. They are found mainly in the white matter but can also occur in gray matter. And very important point you must remember, which is your MCQ as well, that the glial fibrillary acidic protein is found in abundance in cytoplasm of fibrous astrocytes. So next type of the cells are the ependymal cells. These ependymal cells are simple cuboidal to columnar cells that line the fluid filled cavities, that is the ventricle, the central canals of brain and the spinal cord, and they secrete the CSF. These are ciliated to help circulate CSF, and where they are secreting the CSF, they help in formation of the arachnoid granulations. So ependymal cells form epithelial membrane lining cerebral cavities and central canal, and they produces the cerebrospinal fluid. So this is a table which can show you the differences between different types of the cell. The astrocytes, large cells with numerous cell processes in contact with neuron and capillaries. Their function is help to form the blood-brain barrier. They regulate tissue fluid composition, provide structural support and organization to CNS. They replace the damaged neuron and assist with the neuronal development. Ependymal cells are simple cuboidal epithelial cells lining cavities in brain and spinal cord, and they have function in secretion and circulation of the CSF. Microglia, as I told you, smallest of the cells, they have phagocytic in action, oligodendrocyte forming myelin sheath in central nervous system, Schwann cells myelin sheath in peripheral nervous system, and satellite cells are cuboidal cells which are present in the ganglia, and they regulate the nutrients of the cell bodies in the ganglia. So here I complete my today's class. These are the references: Basic Histology by Laikus and Siddiq, the Histology text and atlas by John Quera, and Histology text and atlas by Michael H. Ross. Thank you very much, dear students, for your attention for the today's class, and we will meet in the next class. Till then, Allah Hafiz.